Buenas tardes a todos y a todas. Bienvenidos al seminario institucional. Hoy tenemos presentación doble. Tenemos a la doctora Carla Sedano, a quien muchos de ustedes conocen. Ella nos va a dar una breve presentación de inicio. La doctora Sedano es doctora en Ingeniería y Ciencias Aplicadas. Actualmente trabaja en el Instituto de Energías Renovables. Y bueno, pues ella es, además de una feminista a quien admiro mucho, pues tiene una larguísima trayectoria en el International Women's Forum y mentora de la Global Women's Network for the Energy Transition. Y es actualmente responsable del Laboratorio de Innovación y Futuro del Instituto de Energías Renovables y coordinadora de la Red de Energía Solar y consejera de Voz Experta. Ella nos va brevemente a hablar sobre un proyecto que pues yo también espero que los convenza para participar, y una vez después de 5 minutos con la doctora Sedano pasaremos a escuchar al doctor Scipioni.
Muchísimas gracias. Subo para explicarle eso, ¿no? Gracias, Laura. Gracias a la comunidad del IBT y espero, sí, como dice Laura, convencerles. Les vengo a platicar justo de pobreza energética en el contexto universitario y en México. Y muy rápido, todos sé que hemos escuchado la transición energética y lo que estamos buscando en el instituto desde hace 6 años es hacer investigación para que esa transición energética sea justa. Es decir, que no se sigan replicando los modelos económicos capitalistas hegemónicos donde una persona o entidad vende la energía para que las demás personas la compremos. Y esto solo se puede lograr cuando tenemos esta lógica de la justicia energética, la justicia climática y la justicia ambiental embebidas entre sí mismas. En ese sentido, la justicia energética, para alcanzarla, tenemos que combatir la pobreza energética. Les digo muy rápido cómo definimos en los puntos académicos la pobreza energética. Es un concepto que se empezó a trabajar en Inglaterra hace unos 20 años y hoy día ya es un concepto de uso común en todo el mundo. La pobreza energética se mide a nivel hogar y es la falta de servicios de calidad energéticos en los hogares. Lo que sea que eso signifique, calidad la puede definir cada país, y este es un tema de investigación que es multidisciplinario porque tiene mucho que ver con la percepción y la vida de las personas, que es ahora lo que hacemos, trabajo multidisciplinario, donde la energía es un medio para alcanzar el bienestar y no es un fin. Y el estudio que estamos haciendo con Jessica, que es mi estudiante, es un estudio para evaluar la pobreza energética, pero en la vida de las juventudes. La pobreza energética, por supuesto, que afecta a los espacios rurales, afecta a los entornos periurbanos, nos afecta a muchas de nuestras personas que convivimos. Hay gente que considera que somos pobres energéticos si no tenemos energía limpia, de verdad, de calidad, y pues no tenemos nadie en México porque toda la energía que tenemos es mayormente sucia, porque así se genera en nuestro país. Pero siendo mucho más objetivas, quienes padecen más la pobreza energética son las mujeres, las comunidades vulnerables y las juventudes también. Y un sector bien interesante que estamos estudiando y que se empezó a reportar en Australia y Nueva Zelanda, que son especialmente dañados por la pobreza energética, son las juventudes universitarias. Todo este estudiantado foráneo que tiene que tener un hogar cerca de la universidad y someterse a las condiciones energéticas de quien les renta, de donde se juntan con los roomies, eso es un sector interesante y su desarrollo y la correcta, vamos a decir así, manejo de su pobreza energética es algo que estamos estudiando. Por eso quisimos venir a platicar con ustedes para invitarles a que nos ayuden en este estudio. Primero, queremos entender la pobreza energética a nivel educación superior, que es desde undergrad y posgrado, a todo ese espectro. También queremos evaluar cuál es el impacto de esa pobreza energética, tanto en temas de salud, como pueden ver, de su educación y también de su autonomía del tiempo libre. Eso es muy importante porque es calidad de vida. Y por último, queremos, por supuesto, habrá entrevistas que se harán con algunas personas. Les invitamos a que se contacten con nosotras y con nosotros, porque muchas veces uno renta o está en un espacio donde viven y no toman en cuenta las condiciones energéticas hasta que ya están viviendo en ese espacio. Entonces, si es de mala calidad la instalación eléctrica, si tienen que restringir sus consumos o la forma en la que viven, es un tema que es importante para los estudios de investigación sobre vulnerabilidad y pobreza energética. Y bueno, ahí está el QR. De todas formas, les vamos a mandar un enlace para pedirles que hagan una encuesta. Es una encuesta rápida, está validada por el Comité de Ética del Instituto y de la Universidad de Birmingham, porque antes de tener comité de ética en el IER, teníamos que buscar comités de ética con los colegas que estábamos trabajando, y estos estudios lo hacemos con mucha colaboración inglesa. Entonces, es una encuesta, creemos que es breve, en 15 minutos se llena y nos ayuda muchísimo. Es totalmente anónima, no pide ningún dato, sí algunos datos sobre género, que también cuidamos mucho cómo lo preguntamos, sobre edad y sobre en qué espacio escolar están, pero les agradeceremos mucho si nos ayudan a contestarla, porque será la primera publicación de Latinoamérica sobre el tema. Se le dará su agradecimiento, por supuesto, a la comunidad que nos apoya, pero les quiero decir que en estos temas sí estamos generando conocimiento de frontera a nivel no solo latinoamericano, sino internacional. Entonces, muchas gracias y abro el espacio para la conferencia magistral del día de hoy. Gracias y estamos para servirles en este correo.
Gracias, Carla. Y yo voy a hacer una pregunta que tal vez muchos tenemos juventudes, son ¿hasta qué edad podemos llenar tu encuesta? Si eres alumno de doctor, si eres postdoc, ya eres viejo. Okay. Entonces, está abierto para todas aquellas personas que sean estudiantes o alumnos aquí en el instituto. Gracias, Carla. Y bueno, sin más, quiero darle las gracias al doctor Lorenzo Scipioni por aceptar venir a dar el seminario institucional el día de hoy. Me paro enfrente de ustedes representando al Consorcio de Ingeniería de Bioprocesos y Nanobiotecnología, que en nombre de Tonatiu y el mío, que invitamos a Lorenzo, y agradecer a Dan el que pues nos lo haya presentado, y ya tenemos planes de cómo vamos a conquistar el mundo con él. El doctor Scipioni es investigador postdoctoral en la Universidad de California, Irvine, y me decía Adán, mejor invita al jefe. Dijo, no, yo no quiero al jefe, quiero a quien está haciendo las cosas y está en este momento joven y con muchas ganas, entusiasmo, y espero más fácil de enamorar con nuestros proyectos. Él hizo su doctorado en la Universidad de Genoa en Italia, es italiano. También tiene una maestría y la licenciatura en la Universidad de Pisa en Italia. Tiene 18 publicaciones, las que lista aquí están en Nature Methods, Nature Communications, PNAS, Nature, Nature Cancer, más Nature, y ha recibido financiamiento de un gran número de instituciones internacionales. También quiero resaltar su participación en la formación de recursos humanos. Esto me llamó mucho la atención en cuanto a la supervisión de estudiantes que ha hecho durante su carrera académica. Y también quiero resaltar que recibió el Gregorio Weber International Prize in Biological Fluorescence este año. Felicidades, Lorenzo, por eso. También tiene una patente y bueno, pues también ha ganado una serie de premios en distintas concursos en cuanto a la generación de imágenes. Sin más, le doy la palabra a Lorenzo. Muchas gracias por aceptar nuestra invitación.
This is work? Okay. Can you hear me? Yes. Okay. So first of all, thanks so much, Laura and Tonatiu, for the invitation. It's been a great experience so far. It was actually my first time in Mexico, and it highly exceeded my expectations. So thank you, and thank for all of you for participating. What I want to do today is doing sort of a condensed version of what I did for mainly my postdoctoral studies, and a little bit of what I would like this to do going forward. And so it's a bit of a highly encompassing title. But I want to start from our study system, which is breast cancer. So breast cancer is a very present disease. It accounts for a quarter of all cancers diagnosed in women, and there's been over 2 million cases worldwide in 2020. So it's a very present disease and has different stages. And it actually has a quite high survival rate when it is, get a pointer here, when it is localized or it hasn't spread as much. So that's why also early detection is important. So do your exams. And then, but then when the cancer becomes metastatic and it spreads to other tissues far away from the main tumor mass, it becomes very lethal, actually, when the survival rate drops to lower than 30%. So why the cancers undergo metastasis, and why do they form secondary tumors? Why do they escape therapy? Can all sort of be reconducted to a main mechanism that cancer has. This is true for cancer in general. This is also true for breast cancer. Is a property of cancer cells that's called metabolic plasticity, which is the ability of cancer cells to adapt to changes in the environment and change their phenotype to overcome what other cells see as limitation and keep their proliferating and migratory phenotypes. So, for instance, in a tumor mass, you can have, that leads to a lot of like cellular diversity in the main tumor mass. So you may have different expression, for instance, of 3 common markers for breast cancer that are the estrogen receptor, androgen receptor, and HER2, which are the way that is traditionally used to classify breast cancer. Although, in particular environments, these cells change both their protein expression and their phenotype, and they start invading the body of the host. So you can have different phenotypes, you can have highly proliferating cells, you can have metastatic cells, and you can have chemoresistant cells, right? And in particular, a phenotype that shows none of this protein overexpression. It's called triple negative breast cancer. It's very aggressive and has been shown that it escapes therapy quite a bit. Plus, what the cancer becomes once it is undergoing metastasis also changes the efficacy of these therapies. And on top of that, we have environmental properties such as the pH of the environment, the availability of nutrients, and fatty acids, for instance, that have been shown to correlate with poorer outcome. So what are the current methods to tackle this heterogeneity in cell behavior? You might be familiar with omics technology. For instance, in this case, I make an example with single-cell RNA sequencing, where you can take a sample of the tissue, separate it into single cells, and do single-cell RNA sequencing in each of the cells that can be reduced to a plot which is lower dimensional. And what you see colored here in different colors are clusters of cells that show similar patterns in gene expression. So this can be used, for instance, for classification, for studying the efficacy of therapies, or the appearance or disappearance of particular phenotypes. Although the main problem of this approach is that it doesn't retain the spatial information from where these cells come from. So the next step in the technology has been to translate a similar type of analysis with single cell resolution. And in this case, this has been done by my colleagues in our institution. You can reach the single transcript level. I identifying different patterns, in this case in breast cancer, at the single cell level throughout the tissue, for instance. This is cell culture, but can be, of course, translated to tissues. Now, the problem is that all of these techniques are actually snapshots, right? So you take the cells at a particular time point, you fix them, you treat them, you break the tissue down and so on, but that's only representative of a specific point in time. So the open problem is not much what is the occurrence of a specific phenotype, but where does this phenotype comes from, right? Because we may want to tackle not the symptom, but actually the cause. So we may want the tumor to stay localized so that we can treat it and keep a very high survival rate that I showed before. So our contribution to this is the development of methods that can actually work in living systems rather than in dead fixed tissues, in order to tackle all of this sort of diversity in live cellular models that are, of course, should be representative of the disease. So starting from a very basic concept, fluorophores. We use fluorescent microscopy, and some of you might be familiar with this. Is called Jablonski diagram. Is a diagram that sort of models the process of absorption and fluorescence emission in fluorophores. So these two, S0 and S1, are two electronic energy levels, and when you excite in your confocal or epifluorescent microscope with the light of a certain wavelength, you have a transition to the excited state of the fluorophore. You have a fast relaxation in these non-radiative decays, and then the relaxation to the ground state that results in the emission of a photon with a different wavelength. Then it relaxes back, and you can do this process many times to actually get your fluorescence image, pretty much. Now, this process can be characterized by two main characteristics. One is you're probably more familiar with this. This is the emission spectrum of the fluorophore, which is the distribution of the wavelength that the photon can have for a specific fluorophore. And there's another property that's called the fluorescence lifetime, which shows the distribution of, let's say, the speed of this process. This is less common as a method of characterizing fluorescence, because it's pretty fast. It's typically in the order of the nanoseconds. So you need the specific electronics, and it's a bit more complicated to achieve. So why am I telling all this? I'm telling all this because cells with normal detectors are pretty boring and gray, because all this information is actually integrated over very large spectral bands, and it doesn't include also the lifetime information typically. So what we did was introducing a diffraction grating that would split the light by color. And then instead of collecting with a single detector, we collected with an array of 32 detectors, where this one on top will collect the light in the UV, blue, and this one will go all the way to the red, infrared. And then we made each of these detectors capable of acquiring the lifetime information. So at this point, one image is not just a single image, but is actually a stack of 32 images, each of which is relative to a narrow spectral band. So you can actually see that here you have different structures that appear in the red, in the green, in the orange, and in the red. And in fact, we can actually use this information to color code this image to get an actual color image. So if you were a teeny tiny person standing next to the cell, you would see it fluoresce these colors. And then you can immediately see that you have multiple species in your sample. And taking into account all this information, we can separate them in an image of, in this case, tubulin, mitochondria, and lysosome. All of this is done in live cells. And actually, each of these images is a lifetime image that you can use for further multiplexing or for sensing or other application that I'll show in a little bit. Now, the problem we faced is the analysis of these data sets, because you can, just to give you a scale, instead of getting one image, if you take an image with this setup, it's the same size as if you were to take roughly 8,200 images. So it's a lot of data, and it takes a long time to process. So how do we process this? We use an approach that is called the phasor approach. And I'll show you here an example for the lifetime, and then I'll show you how this changes for the spectral emission. So these are the formulas, but don't care about them too much. I of t is the decay. And in the case of a pure exponential, we can write it as a formula. We can solve this system and get a locus of points, which is basically this semicircle here that you see in black. And then whenever you have a lifetime that is a single exponential, as you can see here, if the decay is very fast, it will be on this side of this plot, and it will go counterclockwise until the decay becomes very, very, very slow. Why are we going through all of this problem? Mostly because we can, from these two parameters, we can directly get the lifetime information, for instance. And then computationally, this is very advantageous because it can be implemented with fast Fourier transforms. This is useful because you can process the whole image, all of the pixels at the same time in any dimension, in one single operation, instead of doing basically a fitting in every pixel, which is very slow. So this can actually accelerate the computation around 3 or 4 orders of magnitude. So you can do in less than a second what before was done in 15 minutes. And this is without GPU acceleration, which of course breaks the timing down even more. Other important properties of the phasor is that if you have a combination of lifetime, which of course usually happens in biological samples, you will have, depending on the amount of photons that are emitted from either species, you will have the resulting combination travel along the line connecting the two. So this is also useful computationally because it's very fast to see how many photons come from one species or the next. That's how the images that I showed you before were mixed, for instance. And the same approach can be applied to spectra. So if you have a spectrum that goes from a blue emission to a red emission, you will have a change in the angle in this plot. And if the spectrum gets broader and broader, it will go more toward the center. So if you consider this in polar coordinate, you will have immediately an idea of how the spectrum would look. Linear combination also work in this space. And in this case, this is a mixture of 3 species, which will have the resulting black spectrum. And depending on the fraction of the spectra that compose the final spectrum, that will have a different position in the phasor space. So all of this led to this publication, where basically in each point of an image, this is a section of a root of a plant, the lily of the valley. And then each point, this is just an intensity image, and each pixel is now not only just whatever number of photons are emitted by all of the fluorescent species, but it's a matrix that encodes all the photophysical properties of all the fluorophores in that specific point. And you can use it, for instance, as a contrast method, and you can see the different structures are highlighted if you use lifetime or spectrum as a method of contrast. And that is it for the plants, let's say. Now, going back to the cells, what we were interested in is looking at as I mentioned, we are interested in something that would tell us what the cells are actually doing in living samples. So we are interested in a class of fluorophores that are called solvatochromic dyes that change their photophysical properties when they interact with the system. So for instance, this fluorophore is called Nile red. You might have heard of this. So instead of having just the canonical Jablonski diagram that you have here, you can also add a different mode of relaxation that has to do with the interaction with the solvent, for instance. In this case, Nile red is whenever it interacts with something that is polar, that has a dipole, it will give it some energy, right? So the more polar is the environment, the more shifted will be the spectrum. So what is the most polar thing that is present in a biological sample? Water. So basically this one can be seen as a probe for water content, for instance. Depending on the spectral emission and the lifetime information, it will be, you can tell how much water accessibility you have in that specific point in your sample. This typically localizes in membranes and lipid droplets. So for instance, you can see how much fat is stored in the lipid droplets or what is the viscosity of the membranes, because that also correlates on how disordered is the double layer and how much water can access it. And there's also another class of probes, the diaminonaphthalene probes that do a similar thing. So they're actually shown with the same model, right? These two in particular are a very similar subcellular localization. So what we wanted to do is that if we can see what is the photophysics of these probes with our method, can we see if their mechanism is actually the same or not? So this is an image of how they look like, except for the spectral differences. One is more in the red-orange, one is more in the blue-green. But basically, this one is Lordan, and this one is red. You see that they stain more or less the same structure. So you have these round structures here, and here are the lipid droplets, because of course you have less water access when you have balls of fat. And then you have membranes that appear more green, or in this case more red. They're more red shifted. The normal way, let's say, to characterize the spectral emission of this is called generalized polarization. I won't go into the details of this, but basically the redder it is, the less access to water you have. So you see how much the lipid droplets stand out compared to the membranes, and we can identify regions where we have, for instance, of course, lipid droplets, and we have less access to water, more access to water, and so on. Now, with our technology, we can actually look, for instance, at the evolution of the spectrum at the nanosecond time scale. This is called time-resolved area normalized emission spectrum. It's a technique that's more common in vitro samples, but we can do it in live samples at optical resolution. And we can also characterize in this phasor space how it evolves. Why is that important? Because if you see, so these are two probes that are supposed to do basically the same thing, right? Just changing the color, changing the lifetime when in contact to the solvent. But if you see how it evolves, for instance, the lifetime, and depending on the spectral emission, you can see that this one evolves in a curved trajectory, whereas this one goes in a line. And the same is true if you plot the reverse, the spectrum as a function of the nanosecond time. You see this goes in an arch, and this goes basically in a straight line. And what does it mean? So if you remember the slide from before, it means that the Nile red behaves as a two-stage system, whereas Lordan has a transition to at least 3 states. And this is actually in agreement with other theoretical work that shows that actually the barrier of potential between these 2 states is quite high. So it can be approximated to a 2-state system, whereas here is more shallow, so you have a sort of more continuous transition between states. So this is just to say that we can characterize with methods that before were exclusive to in vitro systems inside living cells at the organelle level. This is important to make sure that your dyes are actually doing what they're supposed to. And then you can use this information, as I showed before, to do a mixing, and then you have, and you obtain the lifetime images that then you can use, for instance, in this case, for sensing and so on. Even in challenging conditions such as the small spectral difference in spectral emission between the mitochondrial and the lysosomal dye, which is just 14 nanometers. So there is no way that you can separate this based on emission filters, for instance. So that allows you to pack a lot of fluorophores in the same sample, pretty much. And here we did the same on fixed samples, where basically we show that we have the method is valid even with very low signal to noise ratio. And also here we have very challenging condition because these 2 dyes, this one is 10 times brighter than the actin dye, and the spatial overlap is basically in the whole cell because these structures are very present in the cell. Another system where you use it for is biosensors. So biosensors are a series of proteins that you can express in your cells, and they will report on a particular biological process. In this case, this is a chromatin compaction sensor, where you express the histone H3 and HP1 attached to these 2 fluorophores. When HP1 binds to histone H3 to methylate it, the fluorophores go closer together, they perform energy transfer, and they have a change in spectral emission and lifetime as well. So this is a method to measure chromatin compaction pretty much in live cells, which has been shown to be very powerful to detect different levels of chromatin accessibility, for instance, in different cell types. But it has a problem. So as you can see, how do you get this sensor in the cell? You have to express it to make a stable cell line and whatnot. But the problem is that you are expressing, or in some case overexpressing, functional proteins, like histone H3 is already present in the cell. So if you overexpress it and it's systematically attached to HP1, whose work is to methylate it, you end up with a more compacted chromatin overall, which is an artifact of your overexpression. So of course, the right thing to do would be to select cells with a very low expression level. But of course, as you can expect, the signal will be very poor, the sensitivity will be very low. And in fact, you see a lot of noise in the images. Also because of the contribution of autofluorescence that becomes important if your signal is not strong enough. Now, with our approach, we can separate not only the spectrum and the lifetime, but we can do so blindly. We don't have to assume any fluorescent species in our sample. So that at the same time takes care of the low emission the low photon count, because we are actually collecting all the photons that are possible. Plus, it removes autofluorescence contribution. So this is from the exact same data set, but analyzed with the traditional method and analyzed with the spectral film method. So as you can see, we have the distribution of FRET efficiency, which is the parameter that we use to quantify chromatin compaction. It's very different in the case of when you have access to spectral and film information as opposed to just lifetime information. Taking this a step further, we want to apply to some system that is relevant to our field of study, which, as I mentioned, is breast cancer metastasis, for instance. So since 2D cells, cell cultures are not extremely representative of what happens in humans, researchers have developed protocols to make this kind of 3D cultures that are called spheroids or organoids that are grown, in this case, from triple negative breast cancer cells. What do you do with them? So you generate them as basically a ball of cells, and then you embed it in something that resembles the breast environment. In this case, is a collagen matrix to mimic the extracellular matrix. And after 12 hours, you go from just a single sphere to and they start appearing these protrusions of cells. And these are the metastatic cells. So these are the cells that will migrate in the extracellular matrix and then go form secondary tumors. So these are the cells that we want to characterize and eventually target. So how do we do that? We want to see how the metabolism of the cells that compose the main tumor mass and the metastasis differs. So we label them with two dyes. One is Nile red, which is the solvatochromic dye that I showed before. And another one is called JC1. This is a ratiometric probe that measures mitochondrial membrane potential. So this results in 4 emission spectra. So the two, as I mentioned before, Nile red can be considered a two-state system. So it has 2 main spectra. One is the yellow one that colocalizes with the lipid droplets, and one colocalizes more with membranes, whereas JC1 has a greener spectrum that localize in mitochondria in general, whereas active mitochondria, meaning hyperpolarized mitochondria, are shown in a different color. With this method, we can separate it in 4 images. Each of these images is, of course, a film image, so we can do simultaneously with just one acquisition 4-color film. And then we can combine the information from the spectral emissions of all these fluorophores to look at the metabolic fingerprint in terms of lipid metabolism and mitochondrial membrane potential at the single cell level. So as you can see here, for instance, if you look at here is called lipid droplet concentration, but it's basically how much fat is stored in the cells. You can see a higher fat storage in the cells that invade the collagen matrix, and they're also characterized by a higher membrane potential. We can also use the lifetime information to get information about the another parameter for fat storage and also for membrane viscosity. And we can combine all this information to say how the cells in the metastasis differ from the cells that are just proliferating. So giving hints as to how to target their metabolism specifically. Changing completely the topic, because why not? This is another project that has to do with something that is completely different, which is bioluminescence. So bioluminescence is a process that you see commonly in nature. You can see in algae, in fungi, in insects mainly, fishes as well. And it has been used a lot as a marker for a lot of things, mostly in animal models, because it's typically very weak compared to fluorescence emission. So this is usually like gathering bioluminescence information, relies on very long exposures with camera systems. So this is a system that is completely different from our phasor film, which is based on confocal microscopy. One of the problems with this system is, as I mentioned, the photon count, but this has been made better by a group that developed what are called nanolanterns, which is actually pretty smart. So basically, they coupled the luminophore with a fluorescent protein. So then you have energy transfer between the two, and it uses the quantum yield of the fluorescent protein rather than the quantum yield of the luminophore, which is quite low. So in this case, they created a class of very bright bioluminescent proteins. So we teamed up with a group, the Pressure Lab in UCI, where they were not content of having just brighter samples. They wanted to measure multiple bioluminescent reporters in the same sample. So they came to us because they saw the paper and they was like, oh, that's very nice, let's do it. But actually you can't do it because there's no laser excitation here. Bioluminescence is triggered by a chemical reaction. So what we use a technique that is camera-based, that has been developed in the lab, which basically splits the light in 4 channels, 2 of which the reference channels are kept unfiltered, and 2 pass through very special transmission filters that are not the usual band pass that you have in commercial system, but they're shaped like a sine and a cosine filter. Why is that? So going back to math, I'm sorry, you have like I of lambda in this case, it would be the emission spectrum of our probes, right? So this would be the phasor transformation for a spectrum that has a spectrum called I of lambda. And as you can see here, you have the spectrum that is multiplied by a cosine function and then is integrated. And here you have the spectrum multiplied by a sine function and then integrated. So if you put a sine-shaped filter and then you throw your emission spectrum through it, it will be the argument of this integral, right? But when it reaches the camera is colorblind, so it integrates all the photons. So basically what you obtain is that the image here is the numerator of this equation, and the image here is the reference. So with just 3 channels, we did with 4 because cameras are annoying, so we need the reference from the same camera, but whatever. It's more of a technical difficulty. But with 4 channels, you can directly, optically transform the spectra in the phasor space while retaining the image. You can do it at the single cell level, single pixel level. And it works also with these not ideal spectra that you obtain with bioluminescence that are very broad, they're multi-peaked, they're the worst spectra you can actually work with. But when you transform them into phasors, you can see that they actually separate very well, right? Even though you have, like for these 3 spectra, which are 4 spectra, which are 1, 2, 3, 4, you have quite a lot of separation when you use this method. And in fact, we identified up to 6 reporters in the same image, right? It also could quantify very small differences in the emission in the spectral emission of these probes. So even just changing, not by a lot, the residue that you have in this substrate, you can actually identify what is the substrate that activated the fluorescence. So you can use a further level of multiplexing. And since the probes are bright, this method can work in parallel. It's very photon efficient. You can actually do it subcellularly. So we are actually translating a method that was typically used single color in animals to image subcellular structure in a multicolor fashion. And we did that again, tumor spheroids are our thing. So we co-cultured in the same spheroid 3 different lines expressing 3 different reporters, and we can actually see the localization at the single cell level of these reporters within the living spheroid. So we also have another method that is also orthogonal in a way to fluorescence, where we can identify cell types. And this is sort of to show you 2 of the techniques that we developed that use microscopy to investigate the organization of living cells, both from a metabolic perspective and from a bioluminescent perspective, where actually our collaborators are now linking these reporters to proteins that are triggered by gene expression. So we can actually look at the same time at metabolism as well as gene expression in the same living sample and see how it evolves over time. So we are a bit closer to making a comprehensive system that can tackle this kind of heterogeneity. But still, we are lacking a method to look at how cells transition from one phenotype, metabolically speaking, to another, right? So again, so we have omics techniques, we have spatial omics techniques, and then our question is, can we use all of our systems, our knowledge, our approach to create something that is spatiotemporal, so that it retains the same high dimensionality, where you can identify different cell types based on metabolism, but can do so in living samples in order to track dynamic transitions between phenotypes. So this is the new part, don't tell anyone. And the strategy that we're using for this is targeting organelles. So why organelles? So there is a lot of literature that actually shows that organelle characteristics can be used to discriminate, for instance, between different phenotypes of cancer cells. As I mentioned before, like lipid metabolism is very different between metastatic and proliferating cells, for instance. And also, for instance, in neural stem cells, it can be used to identify proliferating cells and lysosomes. There is a very interesting concept in stem cell biology that is called organelle inheritance. So multiple studies have shown that when the cells undergo asymmetric division, during mitosis, the organelles are actually sorted between the 2 daughter cells. And actually, the characteristics of the organelles that will belong to the daughter cell that is differentiated are different from the characteristics of the organelles that will belong to the stem cell, right? So that's also interesting. And plus, of course, mitochondria, because mitochondria do everything. And this is an example of our approach that's based on organelle phenotyping, where we use multiple dyes that are not only organelle specific, but are also environment sensitive. So we can get information related to morphology, number, size, organization, but also function. So we can get, for instance, pH of the lysosomes or membrane potential for the mitochondria. We can get the amount of fat that is stored in lipid droplets and so on and so forth. So in this case, this image is by eye, you can see probably 2 colors, but it's actually 4. Because with our techniques, we can actually pack a lot of fluorophores that have similar spectral properties in the same image. And here we do a single excitation to get a 4-color image. And after a mixing, we can actually localize where are the mitochondria, the lysosomes, the lipid droplets, the other membranes, and get information about their characteristics. In an approach similar to RNA sequencing, we pool all these organelle characteristics in a list of features that then we apply dimensionality reduction, and we get this sort of UMAP, right? Where each point is a cell, and the position in this UMAP is characterized by their organelle phenotype, let's call it. So for instance, cells at the bottom of this plot are characterized by a higher fat content. And as you can see, plus other organelle characteristics. So you can have lysosomes with a more acidic pH that are linked with a higher recycling capability or higher membrane potential, which correlates with the higher production of ATP through oxidative phosphorylation, for instance. There is a phenotype that is associated with oxidative stress, where you have depolarized mitochondria, very acidic lysosomes, and then you have had other high membrane potential phenotypes that are clustered here. And you can actually get very specific pathways such as autophagy, right? Because it's still an imaging approach. So you can literally see the autophagolysosomes because they become these huge, very acidic structures, right? So you can have a sort of like overview of what are the different metabolic phenotypes of a specific cell culture. In this case, these are neural stem cells of the same cell line in growth conditions. So you would expect to have that all of them have the same phenotypes, but actually they are extremely different from one another. And being a method that is applicable to living cells, you can actually see how these phenotypes evolve, or for instance, what do they like to differentiate into, right? And pick only the phenotypes that you're interested in for whatever application. So an example of that is with a simpler system, which is PC12 cells, which is a cell line that can be induced to differentiate in a more neural-like phenotype. And in fact, if you get many of this in differentiation medium, you will get two main clusters, the blue and the red, which are associated with differentiated and undifferentiated cells. So what we did at this point, again, because we can use this at in live cells, we did a 24-hour acquisition of this sample. And we went looking for mitotic events. And then we started looking at the phenotypes of the daughter cells. So we tracked this daughter cell and this daughter cell, all the while assessing their differentiation state. And we can see that daughter cell 1 actually commits to a differentiated state, while daughter cell 2 moves and stays in the undifferentiated cluster. And you can see here is a bit clearer. You see there is a moment of indecision here, and then you have the commitment to fate. So we're actually capable of identifying asymmetric division events in live samples based on this kind of phenotyping. Plus, we have a lot of other projects, of course, that stemmed from this. We should publish some of this to us, but that's a problem for another day. So we applied it to mouse embryonic stem cell colonies, to embryos. We did 5-color, 16-hour time lapses. Actually, we went up to 32 hours and so on and so forth. And we can see that the cells are happily moving, proliferating, and so on and so forth. We did all the biological controls, of course, for the effect of the dyes on the living cells, and so on and so forth. And this is sort of the state of the art on this part. Let's change topic again. So, based on a similar concept, what I want to show you here is that we can do a similar thing by using multimodal microscopy. So multimodal microscopy is the combination. So what you saw now was basically relying on hyperspectral and spectral film imaging. So it's a single technique. But we had another project that still was aimed at understanding breast cancer heterogeneity, but with a multimodal approach. So going back to the system, we make this breast cancer spheroid, we embed it in a collagen matrix, and we start to see these metastatic events. And we want to show what is the difference between the cells that do invade the collagen matrix, because they actually reshape the collagen to make this sort of tracks in the collagen where the other cells would follow, and how they're different from the cells at the surface, which have more access to the nutrients, and the cells at the core that have less access to nutrients. So to do this, we use 3 different techniques. One is based on film, and actually is based on imaging autofluorescent species, in particular NADH, which can be, and we can discriminate with bound NADH, between bound NADH and free NADH. And the balance between these 2 species tells you whether the cell is relying more on oxidative phosphorylation or glycolysis for energy production. Then we saw from our other studies that lipid metabolism is important. So we went again for the NAD red to look at fatty acid storage or consumption. Plus, we added another label-free approach, which is second harmonic generation, where we can actually visualize the collagen fibers and assess the remodeling. The analysis of this is done through an algorithm that I developed for my PhD. I'm emotionally attached to this because it's my first paper. And you can see, for instance, with a control line of normal epithelial cells, which in 2, 12, and 24 hours, you have little difference in metabolic in metabolism, and we have a slight increase in fatty acid storage. And this is what happens instead in our tumor model, right? So we see the presence of the metastasis, they start forming, and then they start invading the collagen, and we can address metabolism at different levels, right? Because we have images to address their metabolic state, lipid metabolism, collagen remodeling, that are all hallmarks of cancer that we can actually quantify at the same time. And we wanted to see how the chemical composition of the environment around the spheroid changes their metabolic phenotype. So we grew them in and embedded in lower density of collagen, because of course, collagen is one of the things that in the breast tissue changes a lot. For instance, for age or other causes. Fatty acid availability has been shown to be also a risk factor, for instance, obesity and so on for breast cancer. Nutrient deprivation as well is has a similar effect. And then, of course, we have our control cell line. So what we did here is that we actually have the resolution to get these 3 hallmarks of cancer at the single cell level. And we did a lot of experiments with this spheroid, each of which is composed by many cells. So we built a database of 200,000 cells from 171 spheroid that shows what is the metabolic heterogeneity of the cells within the tumor spheroids in these conditions. And then we apply the clustering and we color coded them so we can actually see how they are distributed inside the tumor. So we see, for instance, a population of cells that has the same similar phenotype that clustered together. We still don't know what they are. right? So there's a lot of investigation that still has to be done. But first, we need to publish this because otherwise it's been going for years. And then the point then is figuring out what are these, because like one of the interesting open question is, for instance, what are called breast cancer stem cells. So it's a subpopulation of cells in breast cancer that are shown to have stem-like properties, like self-renewal, for instance, and they have been shown to be involved in like tumor initiation, metastasis formation, chemoresistance, and so on. But each of these has like many protein biomarkers. So this is a project for another day. But then it would be interesting to know if we can use this approach to identify this transition between different states in living tumors. Then this is the thank you part. This work has been, so we mostly work on the method development, microscopy, computational part of this. And then we rely on our collaborators for cool biological systems. So I want to acknowledge the Stem Cell Research Center, and in particular, the director is Professor Eileen Anderson for the neural stem cell part, Professor Ann Kaloff for the embryonic stem cell part, the Atwood Lab for another project that I haven't showed because it's my project, for work on skin, and then the Pressure Lab for the bioluminescence, and this is our group. My PI is Professor Graton and Digman, and then the whole group, plus my son, which is photobombing. And then, and this is pretty much the overview of our path toward spatiotemporal omics, if it will ever exist. We'll see. Thank you very much.
Muchas gracias, Lorenzo, por una excelente plática. Vamos a empezar aquí con Adán y después Luis.
Hola, Lorenzo, aquí abajo. I'm making my question in English. So at the end, in terms of analysis, you end up doing principal component analysis on this hyperspectral data, including FLIM and so on and so forth. I wonder if you have explored the possibility of using artificial intelligence, machine learning algorithms to GOTIP, specifically in deep learning or similar stuff.
So I started playing around with neural networks, mostly for image analysis at this point, but it's definitely once we have probably an orthogonal established method to look at the phenotypes, then I would definitely look into machine learning to couple the two methods and actually being able to identify, for instance, protein expression or gene expression, but in living samples. So yeah, that is like, good look in the future. Thank you. Hi.
Thank you for your talk. Thank you for your talk. I really enjoyed that. I have one single question. So regarding the NADH or NADPH autofluorescence, so how fast is your system? Can we look at the fast changes, for instance, the oscillatory changes in the NADH, NADPH? That would be very nice to
quantify fast. How fast? We're talking like seconds,
I don't know.
Microseconds, or
my question is actually if you can look at the oscillations in the pool of the NADH and NADPH.
So I'm not sure if it was published, but definitely this has been done, for instance, for circadian rhythms. Mm-hmm. So we could look at it, I mean, in terms of analysis, taking an image with this modality takes around a minute, because NADH is not, was not born to be a fluorophore, so it's not particularly bright. So it takes a lot, it takes some accumulation to get a decent enough signal. And it really depends on the magnitude of the changes. So some cells, I mean, in response to like treatments, for instance, the changes are quite big. And I mean, the changes are like cyanide or something very like drastic, let's say. But if you're in I mean, anything in a minute time scale, I think is feasible. But it mostly depends on how different the metabolic state of the oscillation are. So that needs a bit more characterization. Yeah.
Thank you.
Thank you, Lorenzo, for your talk. Really interesting. My question is more about blinking fluorophores.
Excuse me?
About blinking.
Blinking, okay.
Can you integrate the theory for blinking fluorophores using Palm and Storm for this kind of technologies? And then can you do some kind of single particle tracking combined with omicsing?
Yes, it actually has been done. I don't remember which group, but they do use spectral fingerprinting approach at the single molecule level to multiplex Storm or Palm datasets. So actually, the constraint that we set ourselves is the two main constraints is the possibility to do this analysis in high throughput and on living samples. So this actually rules out a lot of methods, including all the single molecule methods, because you need the fixed sample, you need longer exposures, you need the blinking. It's a very specific sample preparation. So in principle, it could be applied, although we are getting more and more aware that we need data sets more in the order of thousands of cells to actually tackle like rare phenotypes or have an idea of the actual occurrence of a specific phenotype in different samples. So having that kind of throughput sort of limits, sort of rules out most of the super-resolution methods, for instance, because they typically need a lot of photons, multiple exposures, and so on and so forth. But definitely, for instance, the dynamics properties, for instance, of the organelles is something that can be used to discriminate. So like how fast they diffuse, what kind of diffusion. But that also implies that you need to record a sort of movie for each field of view, which is not ideal in this case. The data sets are very large. Even with just with only hyperspectral imaging, typically an experiment ranges around 200 gigabytes. If you were to add lifetime, it would be 256 times bigger. So it does limit, if you start taking 10 images, even just 10 images per field of view, that would make it enormous. So we're kind of running low on our drives at this point. And the computation would be really intensive at that point as well. So these are sort of the constraints. So in principle, you can also integrate different imaging modalities and so on. But at this stage, we try not to.
Thank you.
Thank you.
So, hi, Lorenzo. There is a question from the virtual world from Christopher Wood. It's about phototoxicity and the duration of experiment.
Yes.
So the question is, in the 60 to 30 hours time-lapse experiment, how do they control phototoxicity? How frequently was the experiment? And he develops, on the time domain confocal spectral film, if they are collecting, say, 1,000 photons per pixel, then I will have thought that the cells will get very stressed quite fast.
Yes, so that is the first thing any of our biological collaborators ask is the toxicity. So behind all the fancy colorful images that I'm showing, there is a lot of troubleshooting, because as you can imagine, for doing this kind of analysis, you need a dye that has a fast uptake, that it stays in the cells for hours and hours. It doesn't bleach, it doesn't kill the cells, it doesn't change the phenotypes, and so on and so forth. So the first thing that we do after we select our subpool of cells that have all these requirements is doing an assay that is called MTT assay or XTT assay, or those kind of methods that are basically colorimetric assays that give you a viability parameter that takes into account proliferation, metabolic changes, cell death, and so on. This is as comprehensive as we've gone so far. And we show that basically with the dyes that we're using at the concentration that we're using, the cells are 100% viable after 2 days of incubation with the dyes. Unfortunately, some of the dyes have to be used at too high concentration, so we had to rule out a lot of them because they were interfering with the cellular processes. But for instance, it also depends on the method of labeling of the fluorophore. So for instance, if you're familiar with mitochondrial dyes for live cells, for instance, there are some that are that localize but do not bind covalently to the mitochondria, while others do. Typically, the ones that bind covalently are also more phototoxic, more cytotoxic. So we had to rule those out as well. there is a bit of trial and error because, of course, until you tested them, there is no database, let's say, of what is good, what is bad. So that was a bit more difficult to troubleshoot. But the ones that we selected are actually capable of tracking cells over multiple hours without cytotoxicity. For phototoxicity, because of course laser exposure can produce oxidative species and then kill the cells. For checking that, so we usually run the overtime experiments with a control. So there is no gold standard method for checking this, but the consensus is looking at proliferation. So if you have a sample that over 16 hours increases the cell number by 1.5 times, twice the cell number, and the trend is replicated in the sample with the dyes, then you don't expect with those imaging conditions to have phototoxicity. It's a bit trickier to get statistics, especially, but yeah, that's pretty much how we check for toxicity. Yes.
¿Alguien tiene alguna otra pregunta? Donatiu?
Thanks, Lorenzo, for a great presentation. So in this spheroids, you will obviously have a very important ingredients of environmental variables, pH, the carbon dioxide concentration, dissolved oxygen. So in order to decouple the actual effect of the environmental parameter versus the inherent cellular characteristic, you would have to control very finely these variables. Do you have a way to control that in these complex systems?
What variables specifically?
For instance, pH or dissolved oxygen.
Oh, I see.
Yeah, not only measuring them, which is great, what you're showing us, but to decouple them, you'll need to have a very precise control.
So actually the gradients in nutrients, pH, and so on are what triggers the change in the phenotype. So it's the whole reason why spheroids are a representative model of the disease and 2D cell cultures are not, because 2D cell cultures have the same, there's no environmental difference between any cell right? Because they're all exposed to the same amount of nutrients and they're all at the same pH and so on. They share the same chemicals. But in a 3D organization, for instance, the cells at the center have fewer nutrients. They typically undergo necrosis. And then you have a layer of proliferating cells that they're not completely exposed to the nutrients, and then you have the cells that are totally exposed to the nutrients, and they get the chemical signaling from the proliferating cells, and they are in contact with the collagen matrix. So there's also a mechanosensing effect, and that's what induces the changes in the phenotype. So this is actually what it's a way to recapitulate the heterogeneity in the, I don't know if I answered the question.
Yes, but within the population, there's still an inherent heterogeneity at the cellular level, yes, which is independent of the environmental variable. So,
yes, so we don't control for that, we characterize it, basically. Yeah, so we don't want a 3D culture that is perfectly layered. Let's say we want one that is representative. So if there is a specific phenotype that arises because by chance you have more cells or they're more packed or there's more tension or something, we want to know that this is a possible phenotype that can arise, especially if that turns out to be a metastatic phenotype in the end. So yeah, we don't control.
Muchas gracias, Lorenzo, por una excelente presentación.
Yes. Gracias a todos.